name is Oliver Fien. I'm director of the West Coast Metabolomics Center. It's my great pleasure to welcome this month's invited speaker, Professor Yang-Yu Liu. He is a theoretical physicist by training, so somebody we all have the utmost respect. Theoretical physics is hardcore. But he has expertise in analytical calculation, modeling, and data analysis. So he received his PhD in physics from University of Illinois at Urbana-Champaign in 2009, and then he did a postdoc. So first, he went to the Center for Complex Network Research from 2009 to 2012. He looked at controllability and observability of complex networks, and had a cover story in Nature and a cover story in PNAS and whatnot, and also in Reviews of Modern Physics, which is really a high reputed journal, by the way, in the realm of physics. And in 2013, he joined the Channing Division of Network Medicine at Brigham and Women's Hospital in Harvard Medical School as a faculty. So that's now 12 years ago. And since then, he has dedicated his efforts to exploring complex microbial community, specifically for the human microbiome, including community ecology and network science and control theory and machine learning. And his papers have been published in Nature, Nature Methods, Nature Machine Intelligence, Nature Communications, and so on and so forth. So we are very honored to have Professor Liu with us today. Professor Liu, the floor is yours.
Thank you very much, Oliver. Let me share my screen. So thank you very much for the introduction. Hello, everyone. It's really my pleasure to speak in this webinar. Many thanks to the organizer for the invitation. I look forward to sharing some insights on the application of deep learning for microbiome-informed precision nutrition that I hope will be valuable and stimulate further discussion today. First of all, let me show you a cartoon. Not sure if you have ever seen this before, since I'm not I'm not visiting on site, so I cannot see how many people raised hand. And also sorry about the bad word. And I promise you, this is just the first time you see this word in my slides. And I would like to re-annotate this cartoon based on my own experience. I studied physics for about 13 years. From my undergrad study on low temperature physics to superconductivity study for my master degree, and then disorder system for my PhD. Then I received postdoc training in Professor Laszlo Barbasi's lab in Northeastern University, working on control theory and complex networks. When I established my own lab back in 2013, I got fascinated by the field of microbiome research. And there are so many interesting questions in this field, and I also contribute a little bit to this field. My microbiome research integrates tools from different disciplines, including network science, ecology, control theory, and machine learning, with the aim to address fundamental questions in the human microbiome and their potential implications in the prevention and treatment of infectious disease such as C. diff infection and COVID-19, and sometimes mental disorders such as post-traumatic stress disorder or PTSD, and the promotion of healthy aging, and the very recently very hot topic, precision nutrition. My motivation of working on precision nutrition actually is due to a very painful personal experience. On a scale of 1 to 10, I think my pain scale was 10. And there was no position in which I was comfortable. And it happened at midnight 4 years ago, and I had to call 911. And that's the first time I called 911. The operator sent ambulance and drive me to the ER. Initially, I thought my son kicked me and broke my ribs because I co-slept with him at that night. But that's impossible because he was only 5. And my boy loved Kung Fu Panda. He watched all the Kung Fu Panda movies, but he never learned Kung Fu, and he was only 5. So cannot be caused by him. So can you guys guess what happened to me? Well, kidney stone. What else?
Yeah.
It has been, excuse me. It has been said that passing a kidney stone is the closed situation a man will get to experiencing the pain of labor and childbirth. So after I passed the stones, I loved my mom and wife much more than I did before. And that's just one consequence of this unforgettable event. Also, after the ER visit, I made an appointment with my primary care physician who gave me something like this. I usually respect my primary care physician very much, but not this time. I have to say this kind of a very generic diet recommendation is not very scientific. It doesn't take into account the highly personalized biological and the lifestyle characteristics, for example, our microbiome, which is very personalized. Thanks to the big efforts of the Human Microbiome Project, now we know that for any given body size, we can never find two individuals share exactly the same species collections and abundance profiles, as you can see from this, those vertical bars represent microbial combinations of different subjects. In fact, our microbiome is so personalized, it can serve as a microbial fingerprint. Based on this PNAS paper, we can identify you from your stool samples generated 1 year ago with very high accuracy. And this notion of a microbial fingerprint was further established in this Cell paper a few years ago. So perhaps you all heard of those two statements: you are what you eat, you are what you think. Now we can come up with a third one. I don't want to say the word, but you all know what I want to say. This highly personalized microbial combination renders the design of generic microbiome-based therapeutics very challenging, but it also makes the diet recommendation and precision nutrition more interesting. Before we talk about precision nutrition, let's briefly review the historical evolution of dietary patterns. As you can see from this roadmap, in the past few hundred years, from the discovery, isolation, and the synthesis of single nutrients to the exploration of the complex biological effects of dietary patterns, nutrition science has shifted and advanced in complex thinking. In the past 100 years, different dietary patterns have inspired a great deal of scientific research related to chronic disease. And those specific dietary patterns include ketogenic diet, Mediterranean diet, high fiber diet, the Dutch diet, plant-based diet, etc. And those dietary patterns have been demonstrated to be quite helpful in preventing certain diseases. For example, research has constantly shown that the Mediterranean diet is effective in reducing the risk of cardiovascular disease and overall mortality. In 2023, we also published a paper showing, showing that individuals who adhere more to Mediterranean diet tend to have less PTSD symptoms. And this finding was covered by NBC News and many other media. Nowadays, we're often talking about multi-omics approach to build precision nutrition strategies. So what is precision nutrition? Well, according to the 2020 to 2030 strategic plan for NIH nutrition research, precision nutrition is a framework canvassing a wide array of features, including genetics, dietary habits, eating patterns, circadian rhythms, health status, socioeconomic and psychosocial characteristics, food environments, physical activity, and the microbiome. And there are multiple aims in precision nutrition research. First, we aim to be more precise in measuring diet and nutritional status, for example, using biomarkers or smartphone apps. Second, we aim to be more precise in understanding the biological mechanism of diet. Third, we want to be more precise in dietary recommendations at the population level. For example, the types of fat and carbs, daily intake limits for sodium, sugar, and saturated fatty acids. And finally, we want to be more precise in giving dietary advice at the individual level. For example, using AI to prescribe personalized diet to achieve the so-called personalized nutrition. I'm personally most interested in the last aim, like the personalized nutrition. Despite some criticism, actually very, very harsh criticism on the evidence of personalized nutrition, and also the noise due to excessive promotion made by some companies. This trend, moving from generic dietary guidelines to personalized diet recommendations, is actually quite natural and unavoidable. After all, we are so different. Due to highly personalized biological and lifestyle characteristics, individuals may have very different metabolic responses to specific foods and nutrients. In particular, our gut microbiome is highly personalized and plays very crucial role in our metabolic response to foods and nutrients. So I do believe we need personalized diet recommendation. We just need to do it right and be humble, do not oversell it. In this slide, I list a few methods we recently developed, which we hope can facilitate precision nutrition research. In the first paper, nutrition redundancy paper, we proposed a new concept, the so-called nutrition redundancy, which represents the average nutrient similarity or overlap of two randomly chosen food items in any diet assessment. We find that a high nutrition redundancy is associated with lower risks of cardiovascular disease and type 2 diabetes. So this measure may be helpful for us to be more precise in giving dietary recommendation at a population level. And CNODE is a deep learning method that can be used to predict microbial composition based on the species presence-absence pattern. As a follow-up of CNODE, MNODE is another deep learning method. It can be used to predict the metabolomic profiles of microbial communities from the species composition. MLP is another deep learning method for the prediction of metabolite response using baseline microbiome data before the dietary intervention. And this could help us prescribe personalized diet to achieve optimal metabolic response. And metric is a deep learning method that can leverage gut microbiome data to correct recall bias in the nutrient profiles computed from self-reported dietary assessment, such as 24-hour dietary recalls. ODR is an optimization algorithm that develops personalized diet plans based on the existing dietary plan and calculate healthy diet scores with the goal of optimizing an individual's healthy diet scores. MPDR is a deep learning method that develops personalized diet plans based on the existing gut microbial combination, aiming to achieve optimal gut microbial combination. For the interest of time, today I will present you those three methods: CNODE, MNODE, and MLP. Among them, I have to say that the last one, MLP, is the most relevant one for precision nutrition, but the three methods are closely related to each other, as you will see. So it'll be better for me to introduce the first two methods before I talk about MLP. Let's begin with CNODE. The acronym stands for Computational Neural Ordinary Differential Equation. I know this has nothing to do with precision nutrition, but what can CNODE do for us? Well, let's consider a microbiome sample you collected from a particular environment or body site. This sample has certain species present, represented by a binary vector, Z. Can we predict their relative abundances just based on their presence? You may say, you're kidding me, this is impossible, and I completely agree with you that if you have only one sample, and you have zero knowledge on the microbial interactions and environmental conditions, this is impossible. But if you have many samples and with some reasonable assumptions, this is actually doable. Mathematically, we need to learn a map, which is a nonlinear function and presumably could be quite complicated. And this map connects the binary vector Z, representing the presence-absence pattern of species in the sample, to the community composition vector P. Since we are talking about combinations or relative abundances, this vector P is living in the so-called probability simplex denoted by the triangle symbol, which means that all the elements in the P are non-negative and add up to 1. To ensure this function phi, or the map, exists, and the problem is mathematically well-defined, we need to make a few assumptions. First is about the universality. Which means that those samples share universal dynamic rules so that the function phi is the same across different samples. For in vitro communities, the universal dynamics assumption is satisfied if the samples were collected from the same experiment or multiple experiments, but with very similar environmental conditions. For in vivo communities, a few years ago, we demonstrated strong empirical evidence that human gut and oral microbiome of healthy adults display very strong universal dynamics, despite quite different host factors such as race, age, BMI, long-term dietary pattern, and stool consistency. Second is about equilibrium, which means that the compositions of those microbiome samples represent at least roughly the steady states rather than transient states of the system. This assumption is natural because if the function phi is not well defined for highly fluctuating microbial combinations. Many observational studies of host-associated microbial communities, such as the human gut microbiome and the oral microbiome, the skin microbiome, indicate that they remain close to stable steady states in the absence of drastic dietary change or antibiotic administration. And then finally, we assume there's no true multi-stability for the species pool or any subset of it, which means that for each species collection Z, there's a unique steady state combination P. Okay. In practice, of course, we don't expect that all the three assumptions can be strictly satisfied. So our ability to accurately predict microbial combinations from species collection is actually a posteriori evidence of the validity of those assumptions. Okay, now you may wonder how to learn the function phi. Well, we turn to artificial neural networks. In the past several decades, different architectures of neural networks have been developed for different purposes. And here's a chart of artificial neural networks up to 2016. I cannot find an updated version of this beautiful chart. And today I don't have time to describe each of the architecture, but I will tell you exactly how we ended up using neural ODE, a relatively new family of deep neural networks, to solve our problem. So let's begin our journey. Our goal is to learn a very complicated function phi. Thanks to the universal approximation theorem, we know that even for a single layer feed-forward neural network with a sufficient number of neurons, it can be used to approximate any continuous function on a closed and bounded subset of a Euclidean space. This is really encouraging, but there's a caveat. The function phi may be very complicated and may require an impractically large number of neurons in this simple network to approximate accurately. In practice, it's common to use more complex neural network architectures, such as deep neural networks with multiple layers, to approximate complex functions accurately. Actually, it has been shown that if you think of a neural network as a function approximator, then the complexity of the approximation function grows exponentially with depth, not width. So with the same number of parameters, a deep and a narrow network has more expressive power than a shallow and wide network. That's why we need a deep learning, not wide learning. And as Leonardo DiCaprio told us in the movie Inception, we need to go deeper, not wider. Empirical results have demonstrated that deeper networks generalize better for certain tasks. For example, when they are used to transcribe multi-digit numbers from photographs of addresses, as you can see here, the test set accuracy consistently increases with increasing number of hidden layers or depths. However, there's another caveat. Deeper neural networks are much harder to train, and there are two notorious problems attributed to the training difficulty of deep neural networks. First is about the vanishing and exploiting gradients problem. This problem is encountered when training neural networks with gradient-based learning methods and backpropagation, which is based on the chain rules in calculus. This involves repeated multiplications of gradients. And we know that if we multiply many small or big numbers, the product might decrease or increase exponentially with the number of the numbers, as shown in these two figures. Fortunately, this problem can be largely addressed by normalization techniques, so it's not a big deal anymore. However, when deeper networks are able to start conversion, a degradation problem might emerge. This means that with the network depth increasing, accuracy gets saturated and then degrades rapidly. Such a degradation is not caused by overfitting, because adding more layers to a suitably deep model leads to higher training error, as shown in this figure. Fortunately, this problem can be addressed by introducing the residue learning framework, or the so-called residue neural networks, aka ResNet. The key idea underlying ResNet is the use of skip connections to facilitate the training of very deep neural networks. As you can see here, the top one is the 34-layer plane network without skip connections. The bottom one is a 34-layer residue network. The two networks share exactly the same number of neurons and parameters. The only difference is that the residue network has the so-called skip connections, as shown in the black arrows. which allow the output of a layer to bypass one or more layers and be directly fed into a later layer. This creates a shortcut that enables the gradient to flow more easily through the network during the backpropagation, improving the training of very deep networks. Okay, but why is it called residue neural network or ResNet? We know that traditionally when building a deep neural network, each layer is designed to transform the input data into a high-level representation as shown in this equation. Okay, so but a ResNet actually explicitly fits a residue mapping. So the right-hand side is the residue. That's why it's called a residue neural network. And this equation can be rewritten as follows. So which can be realized by feed-forward neural networks with skip connections. And the skip connections are implemented as just simple identity mapping that add the output of one or more previous layers to the output of the current layer. And by doing so, the network learns to focus on the residue difference between the desired output and the current output of the network, rather than trying to learn the entire transformation from scratch. So this is a very clever idea. But only 2 years after the invention of ResNets, neural ordinary differential equation, or neural ODEs, or NODE, was developed. The key idea of NODE is to use ordinary differential equations to define the dynamics of neural network. So rather than defining the network as a series of discrete layers that transform input data into output, the neural ODE defines a continuous flow of information through the network, allowing for more flexible and expressive modeling of complex data. This is also a very, very clever idea. Actually, if you can see the two equations, the ResNet equation can be considered as the classical Euler method for solving ODEs. And the equation on the right is nothing but the ODE that the Euler method is trying to solve. Okay. Of course, nowadays, we don't have to use the Euler method to solve any ODEs. Euler method is perhaps the simplest and oldest ODE solver that was proposed by the mathematician Leonhard Euler in the 18th century. And they have since been more than 250 years of development of efficient and accurate ODE solvers. Modern ODE solvers provide guarantees about the growth of approximation error, monitor the level of error, and adapt their evaluation strategy on the fly to achieve the requested level of accuracy. So in the context of a neural ODE, we don't specify the depth or the number of layers. Actually, it's left to the ODE solver, and we don't really care. Based on the success of neural ODE, we developed convolutional neural ODE, or CNODE, by defining the following hidden layer dynamics as follows. Here, the f theta can be any continuous function parameterized by theta, and this function maps the the computational vector to Euclidean space, and this Hadamard product represents element-wise multiplication. For those of you who are familiar with evolutionary game theory, you may realize that this ODE is kind of a general form of the so-called replicator equation, which is the canonical model in evolutionary game theory. And f theta was typically referred to as the fitness function. To solve this ODE, the input is just the species collection, which is the binary vector z. From z, we compute the initial condition of this ODE, and we choose the final integration time to be 1 without loss of generality. Then we integrate ODE to compute the final solution h tau c, which is nothing but the computational vector we want to predict. In this figure, I'm showing you the architecture of CNODE, and here we use the simplest fitness function f theta equals theta times h. Theta is n by n real value matrix. In principle, you can use a more complicated function represented by a feed-forward deep neural network, like a multilayer perceptron, to parameterize f. But in this paper, we just use very simple form for the fitness function. By the way, the ODE in this particular form is really the key to the success of CNODE, because it naturally satisfies two conditions, such that the predicted vector must be computational, and second, the predicted abundance of any absent species must be identically 0 so that we don't have any true positives, false positives. To train the C-node, we need to adjust the parameter theta in the fitness function f theta and minimize the prediction error in a training dataset, which is nothing but a pair of z and p vectors obtained from a given set of microbiome samples. At the end of the day, a well-trained C-node can be used to predict the community composition from any given species collection. By the way, the stacked bars shown in this slide, they are not fake data, but real gut microbiome samples collected from the fruit flies. And as you can see, the predicted microbial composition agree well with the observed one in the experiment. We systematically validated C-node using synthetic data generated by the generalized logical vector model. Then we applied C-node to analyze several real data sets from environmental to host-associated microbiomes, and it works very well. You may wonder, why should we care about C-node? After all, the price to get a Z vector and a P vector is almost the same for complex microbiome samples because we just need to do metagenomic sequencing and get Z and P at the same time. Why should we care about the prediction from Z to P? Well, in a follow-up paper, we show that using a well-trained C-node, we can perform the so-called thought experiments. We can remove any species from any given community, and we can predict the consequence of this species removal. And this helps us a lot to identify keystone species from complex microbial communities in a purely data-driven fashion without using any ecological model or conducting any real experiments. As you may know, keystone species is a very classical and fundamental concept in community ecology. It means that those species, despite their relative low biomass or relative abundance, the removal of them could cause a huge impact on the community. It may cause many other species die out, and the whole system might collapse. So it's quite natural to identify keystone species from microbial communities. Our deep learning-based method is the first computational method that can correctly identify keystone species in a purely data-driven fashion without doing any real experiments or using any ecological models. In fact, you can do whatever you want. You can not just remove one species, you can remove many, or you can add some new species, and C-node can be used to predict the consequence of the intervention. And that's very cool. Actually, later on, based on other deep learning methods, I'm going to show you how thought experiments can be designed to infer food microbe and microbe metabolite interactions, which are very helpful for precision nutrition. Okay, now let's move on to the second method, M-node. Just like in the C-node project, let's consider a microbiome sample collected from a particular environment. Suppose you already have measured the microbial composition using metagenomic sequencing. Can we predict the metabolomic profile of this community based on its microbial composition? Asking this question is meaningful for two reasons. First, we typically don't have access to microbiome and metabolome data at the same time. And we believe that metabolomics is harder and more expensive than metagenomics. You guys can correct me if I'm wrong. So it's desirable to develop a computational method to predict the metabolomic profile based on the microbial composition. Second, such a method could facilitate our understanding of the interplay between the microorganisms and their metabolites through thought experiments, as mentioned before. If you have only one sample and have zero knowledge on the genome scale metabolic network of each species in the sample, making this prediction is impossible. But again, if you have many samples, then we can learn a function that maps the microbial composition to the metabolomic profile. Many machine learning methods have been developed to solve this problem. They all have some limitations and don't perform very well, as you will see later. Based on the success of C-node, we developed M-node. A key difference between the two methods is that in the C-node case, the species collection vector Z and the microbial composition vector P naturally have the same dimension. But in the M-node case, of course, the microbial composition vector P and the metabolite concentration vector Q generally do not have the same dimension. So the original neural ODE framework will not work. Our solution is very simple. we just sandwich the node between two fully connected neural networks. So basically, we introduce some latent vectors. So we transform the input, which is the Markov combination, to a latent vector, and the latent vector will be the input of the neural ODE. The output of the neural ODE will be another latent vector, and we transform the latent vector into the metabolic profile, which is the output of M node. For the neural ODE module, the function f that define the ODE or describe the dynamics of the latent vector is parameterized by a neural network. For simplicity, we just use the multi-layer perceptron with only one hidden layer. And this has the same dimension as that of a latent vector to parameterize the function f. Here, we didn't design ODE to have any particular structure as we did for the CNOTE project. This is because we perform the so-called center log ratio or CLR transformation to both the Markov combination and the metabolomic profile, so that the input of the neural ODE is not computational data anymore. Actually, both the input and output of the neural ODE are latent vectors of a certain dimension, and they are not subject to any constraints. And this M node architecture actually is quite general. Because we sandwich the node between two fully connected neural networks, we can incorporate different types of input data, for example, dietary intake data, to further enhance the prediction of the metabolic profile. Here are some technical details of the M node method. I will skip them. To validate M node, we generated synthetic data using the so-called Markov consumer resource model. Is the very sophisticated model that explicitly models Markov interaction due to nutrient competition and the metabolic cross-feeding. I will skip the details of this model and the validation. Basically, it works and outperforms existing methods, as you can see from the three performance metrics. We then test M node on real data. In this table, I list 5 different data sets from 4 studies. As shown in this slide, across the 5 data sets, M node delivered overall best performance based on 3 performance metrics. Just like the CNOTE method, a well-trained M node can help us conduct thought experiments. For example, we can perturb the relative abundance of a species I and measure how the predicted concentration for metabolite alpha changed, and the sign of the so-called susceptibility of metabolite alpha to species I can be calculated in this way, which can be used to reflect the interaction between metabolite alpha and the species I. Presumably, if this coefficient is positive or negative, it may imply species I can produce or consume metabolite alpha. We validate this idea on the synthetic data generated by the Markov consumer resource model. In this case, we can directly compare the susceptibility matrix with the ground truth consumption matrix and the production matrix in the model. And it's evident that many ground truth interactions are reviewed in the susceptibility matrix. We found that for synthetic data, the susceptibility can predict consumption and production interactions with great performance, with AUC higher than 0.85 for both interaction types. Then we apply the sensitivity analysis to a real data set related to the fecal microbiome and the metabolome samples of patients with IBD. We identified several interactions indicated as purple boxes here that agree well with genomic evidence. For example, this box is called Bacteroides voctus. It contains a so-called biosalt hydrolase or BSH gene responsible for the deconjugation of conjugate primary bile acids to primary bile acids. And this bug is called Lectospirillous bacterium, has a bile acid induced or BAI pathway, which is capable of transforming primary bile acids to secondary bile acids. And this bug is called Streptococcus salivarius, contain no genes related to the bile acid metabolism. So the sensitivity analysis based on a well-trained M node model seems to be quite useful to explore the microbe and metabolite interactions in a purely data-driven fashion without using any domain knowledge. Okay, finally, let's talk about MAC-MLP. This project was inspired by this seminar paper. We know that people eating identical meals present high variability in post-meal blood glucose response. In this 2015 cover story Cell paper, the authors demonstrated that personalized diet created with the help of an accurate predictor of blood glucose response may successfully lower post-meal blood glucose and its long-term metabolic consequences. For the machine learning model, the authors used the traditional method, the so-called gradient boosting regressor, or GBR, to predict the personalized postprandial glucose response, or PPGR, to meals based on individual factors such as dietary habits, physical activity, blood parameters, anthropometric data, and the gut microbiome composition. The stool samples were collected within 3 days of the trial starting date. After being trained on a cohort with 800 participants, the machine learning model gradient boosting regressor, or GBR, was validated using an independent cohort, achieving a Pearson correlation coefficient 0.7 between the predict and measured PPGR values. It's unclear if deep learning can further improve the prediction performance. Now let's ask a more general question. Can we predict the metabolite response to dietary intervention from the baseline gut microbiome composition and the baseline metabolite concentration? So now we don't just focus on blood glucose, but we interested in general healthy related metabolites. Clearly, this is quite relevant for precision nutrition purpose. Previous studies typically use classical machine learning methods such as random forest or gradient boosting regressor to solve this type of question. We wonder if we can do better by using deep learning models. Inspired by the success of MNode, we naively thought we can just apply MNode to solve this problem. It turns out that MNode doesn't work very well in this case. In fact, we found other deep learning models like MLP, multilinear perceptron, do not work well either if we still follow this one-step strategy. So we have to respect the difference of the problem settings. MNode was designed to predict metabolomic profile of a microbial community based on its current microbial composition. Now we want to predict the endpoint metabolite concentrations after a particular dietary intervention based on the baseline data measured before the dietary intervention. So the two problems are fundamentally different. Moreover, MNode performs well when the number of metabolites is large, especially for untargeted metabolomics, typically with hundreds or thousands of metabolites. This is because MNode and other similar methods rely on techniques to reduce model complexity, such as regularization, the encoder-decoder architecture, etc. However, for many dietary interventions datasets, as you will see later, only dozens of metabolites were measured as biomarkers using targeted metabolomics. We believe that in order to predict the endpoint metabolite concentrations very well, we have to first capture how microbial composition changed from the baseline to the endpoint. That motivated us to develop a two-step strategy which predicts endpoint microbial composition using a deep learning model, and then include the predict endpoint microbial composition as a part of the input for another deep learning model to predict the endpoint metabolite concentrations. And we found that we can just use MLP as a deep learning model for each step. And since those two MLPs are coupled, we call the entire model metabolite response predictor using coupled multilayer perceptron, or MAC-MLP for short. In this slide, I'll show you how to train and test MAC-MLP. It's quite straightforward. Here are some technical details of MAC-MLP. I want to emphasize 2 points that are both related to the targeted nature of the metabolome data in dietary studies. First, regarding the data processing. Unlike the MNode project, where we're dealing with untargeted metabolome data, which is computational, so we try the CRR transformation. Here, for dietary intervention studies, we have targeted metabolome data for which we have the absolute about concentrations. That's why we use log10 transformation. Second, since in dietary intervention studies, typically only dozens of metabolite biomarkers were measured, we developed MacMLP based on the so-called over-parameterized MLP with 6 hidden layers, and each hidden layer has more than 2,000 neurons. Over-parameterization can make models even simpler due to smoother function approximation and less likely to overfit. So the over-parameterized models can generate better and more consistent performance than models with less capacity and more carefully tuned hyperparameters. I know this is kind of a counterintuitive, but this type of a modern machine learning practice using over-parameterized model and the classical bias variance trade-off has been recently reconciled in a very nice framework based on the unified performance curve known as the double descent risk curve. And here's a reference in case you are interested in this topic. As usual, we first validated MacMLP using synthetic data for which we know the ground truths. Again, we used the McCubic consumer resource model to generate synthetic data. We found that based on 3 performance metrics, overall MacMLP achieved the best performance. The performance of all the methods can be improved further by including baseline metabolite concentrations as part of the model inputs, especially for MacMLP. Furthermore, we found that a larger training data sample size will improve the prediction performance, which is quite natural. We also found that as the intervention dose increased, the predictive power between including and not including the baseline metabolite concentration shrinks. We believe this is because a larger intervention dose significantly changed the endpoint metabolite concentration away from its baseline level, rendering the baseline level data less useful. Now let's talk about real data. We analyzed data from 6 dietary intervention studies. 5 of them were kindly shared by Professor Hannah Hochschler from UIUC based on her complete feeding trials. As you may know, to conduct complete feeding trials, you need to recruit participants and feed them in a completely controlled manner for all their meals, just like what you will do in animal experiments. That's not easy. I even have difficulty convincing a single person, my son, to eat more vegetables. But Professor Hochschler can make many adults to eat whatever she wants them to eat. So that's amazing. Across all the studies, fecal or blood samples were collected before and after each dietary intervention period for metagenomic sequencing and targeted metabolomics. We found that when the baseline metabolite concentrations are not used in all the methods, MacMLP is better than the other 2 methods, random forest and the GBR. When we included the baseline metabolite concentration as part of the model inputs, it further improved the performance, especially for MacMLP. And MacMLP consistently produced the best performance across all the datasets. The relatively poor performance excuse me, of all the methods on the data from the study that investigated fibers and the fermented food is likely due to that a variety of foods within the fiber and the fermented food category were consumed by the participants at will. So by definition, this is not a completely feeding trial, and we might need a much larger training data for this type of study. Similar as the MNODE project, we can also perform sensitivity analysis based on a well-trained MacMLP. This enables us to study the tripartite food micrometabolite relationship. The idea is exactly the same as that in the MNODE project, so I will not repeat it here. As usual, we first evaluated our sensitivity analysis on the synthetic data for which we know the ground truths, and we find it performs very well. Then we applied the sensitivity analysis to real data. Here I show you the results from analyzing data in the avocado intervention study. This heat map demonstrates the sensitivity values for the top 20 avocado microconsumption interaction in green and the top 20 micrometabolite production interaction in red. Based on this heat map, we also constructed the avocado microbe butyrate triplete graph. The edge widths are proportional to the absolute values of the sensitivity coefficients. All the microbes in the middle layer are arranged from left to right in the increasing order of the edge widths. So, excuse me, this way, species on the right tend to be strong consumers of avocado and strong producers of butyrate. So if an individual has more microbial species on the right, he or she is likely to have a positive butyrate response to avocado. And we found that this particular bug, the so-called Faecalibacterium prosnithizi, or FP, emerged as the most important species for butyrate production in response to avocado intervention. And this finding agrees well with previous studies. So we think a well-defined MACMLP can help us explore more interesting food microbe metabolite relationship and facilitate the design of personalized diet recommendation to achieve precision nutrition. I would like to conclude my talk by a conceptual diagram. The key message I try to convey to you today is that deep learning can facilitate microbiome-informed precision nutrition. And indeed, by analyzing the gut microbiome, we can predict how an individual will respond to a specific food and a diet intervention, leading to more effective and personalized diet recommendation. And this microbiome-informed precision nutrition can be facilitated with deep learning techniques and metabolomics. Finally, I would like to thank all my former and current group members. And I'm proud to say that 21 of them are independent PIs right now, and 7 of them have already become tenured. Also, I'd like to thank all my collaborators and my funding agencies. And thank you for your listening. I'm happy to take any questions.
Thank you. Thank you so much, Professor Liu. That is fantastic. Confusing to people who are not in all the fields that you mentioned, especially not like proficient in mathematics. That was brilliant, and I loved it. But there were, of course, questions here, and then to all the participants, if you have now watched it and you have other types of questions, just put them in, right? Put them into this to the chat so that we can read it. So I'll start from the end, if you don't mind. So there were 2 questions by 2 people, basically saying, look, not all the metabolites that you see in fecal matter are produced by microbes. Some might be produced by the host, meaning from the liver and whatnot, and others might be produced like leftovers from food. How do your methods control for that? Is because that's not microbe related, hence it might be noise in the system. Do your attention layers just forget about those?
That's a very, very good point, actually. Of course, our model cannot distinguish whether these metabolites were produced by metabolites or by the host. I would say this kind of a noise in the problem. That's why the prediction is not perfect.
Okay. So then we have a very simple technical question. What do you think about the differences for microbiome data between whole genome sequencing and 16S sequencing? Which sequencing approach or which data types did you use to train your models for microbiome compositions? Have you noticed any differences between the data types and whole genome sequencing versus 16S? And then follow up, which software did you use for processing? Was it QE2 or Kraken or other types of software like MetaPhi-N? And have you observed any performance differences in those models when using different tools?
Yeah, okay, that deserves another talk. First of all, our method, all the deep learning methods I discussed today, like C-node, M-node, or MACMLP, can deal with both 16S RNA gene sequencing data or whole metagenome shotgun sequencing data. It doesn't matter to those methods. In the MACMLP project, because for most of the dietary intervention studies, my collaborator, Professor Hochschuler, she did 16S RNA gene sequencing. So we used the 16S data. For some other data sets, because we have the shotgun metagenomics data, so we just leverage the metagenomics data. So it doesn't matter to us. Okay, so both data types works for those deep learning methods. Regarding the software we use for data processing, I think the method you referred here are kind of a reference-based taxonomy profiling or metagenomic profilers. And we have another paper showing that almost all the existing metagenomic profilers, they suffer a lot from false positive issues. And we actually developed our own method. It's called Map2B. It's a Nature Communication paper published in 2023. Feel free to check it out. And I also highly recommend that if you have access to the raw sequencing data, you'd better do the assembly and binning rather than rely on any reference database, because very likely if you just do the assembly and the binning, you will identify or discover some novel species that never exist in the reference database. That's my recommendation.
Okay, cool. Frightening to some extent. There was a very simple question. What do you mean by metabolomic profiles? Selecting significant metabolites or also unidentified features associated with microbiomes. So metabolomic data sets and untargeted metabolomics might be compounds that have a name or compounds that don't have a name. We call them features, right? Do you take both in or just the ones that have names?
We take everything. In the deep learning model, we don't care the names. They are just different features.
Okay.
So, yeah.
Yeah. Later on, you showed us names. So.
Oh, so yeah. In the MacMLP paper, in that project, because we are dealing with targeted metabolomics data, of course we know the names.
Yeah.
Yeah.
That leads to some kind of question that I had actually. So the problem is, of course, when you have different metabolomic labs, they have different metabolomic profiles because they use different assays and whatnot. So how do you learn a model and then apply it to something else that had different metabolites? I mean, how do you standardize your data for the M node outputs from the learning? So I don't know. I would understand if everything is standardized, and so you predict metabolomes based on one method, and then you can take it for another one. But how do you validate that? How do you deal with that non-synchronization of the data?
Yeah, if different labs generate different sets of metabolites, for example, right? I guess we have to retrain the model, right? We can't just naively apply a model trained based on the metabolite data generated by lab A to lab B. I know this kind of it'd be a nightmare, but we have to be quite honest. We cannot oversell the model.
Okay, cool. So that is kind of like related to another question. When you generate new methods, it is reasonable to say you need to compare to existing alternative methods, and sometimes you've shown it in your talk. But where do you find good, true ground truth data for benchmarking for these method comparisons?
Can you repeat the question again?
So benchmarking, it's about benchmarking. You have different methods, right? And for benchmarking, you need ground truth.
Yes.
But how do you know the ground truth if...
Oh, I see. Okay, that's a very good point. That's why in many of our studies, we rely on, I think I touched upon this, the so-called Markov consumer resource model. This is kind of a set of ordinary differential equations that can model the very complex interactions between microbes and the nutrients. In the model, we know exactly how the microbes interact with the nutrients. So we know the ground truth. That's why in the validation, the C matrix and P matrix, they are known, they are ground truths. And the susceptibility matrix is inferred from the data, the synthetic data. We can compare this matrix with those two. We can do the calculate the AORC to validate our method. So we always use synthetic data as the benchmarking data because we know the ground truth.
Okay, so you basically generate it first. Okay, that
Yeah,
makes sense.
we generate the data. Yes.
Yeah. So there was a specific question: how much does C-Node improve the results compared to prior neural ODEs? That was in the beginning of the talk. So C-Node improve results compared to prior neural ODEs.
Okay. I guess by prior neural ODE, you mean the vanilla neural ODE, right?
Yeah.
So vanilla neural ODE will not work for this problem because we are dealing with computational data.
Yeah.
And we have to design the hidden layer dynamics in a very special way so that the prediction of this C node is a computational vector. The vanilla neural ODE cannot solve the problem. So this is not comparable. Let me put in that way. So we are the first to set up this problem. We come up with C node. You can argue that C node is a variant of a neural ODE. We leverage the strength of a neural ODE. We modify it a little bit by introducing this hidden layer dynamics so that it can serve our purpose by predicting computational vector. And all the initially absent species, the predict abundance will be exactly 0. So there will be no false positive.
Mm-hmm.
Okay.
So then we have now while we are talking and discussing, there are new questions have arisen from participants that are more generic or general questions. So the first one is, could you share insights how the results and your methods can be actually used for precision nutrition? It seems like currently the dietary advice is still somehow generic, like eat more avocado or less meat or
Oh,
and
no.
No.
Well, so if we go back to the tripartite graph,
Yes.
the key message I want to convey here is that you see this particular bug is called Faecalibacteria proselytici, or FP, is a strong consumer of avocado and also a strong producer of butyrate. We know butyrate is a very beneficial short-chain fatty acid for the host, for the human health. So depending on the presence of this particular bug, the response to avocado, to this particular dietary intervention could be very different. So that's the point, right? So we're not making population level recommendation.
Yes.
The recommendation has to be considering the microbial composition of the participant.
Mm-hmm.
That's the whole point of precision nutrition.
Thank you. Yeah, I get it. So that means we need to sequence those people.
Yes.
The microbiome of those people first.
Yes, the microbiome. Yes, in order to make scientifically technically sound recommendation.
Then we have a question. Could you please reiterate what to use instead if the references to potentially find new species? What to use instead if the references to potentially find? I'm not quite sure I understand the question.
I guess the papers?
By Nushin Mir. Nushin, do you want to tell more? Nushin Mir? I guess you can unmute yourself. Nushin? I am, the Nushin Mir at the very end said, could you please reiterate?
I guess I understand the point. So basically it's about how to discover new species, because I mentioned that if you rely on the existing reference-based taxonomy profilers like metagenomic profilers, you might miss some novel species in the samples.
Okay.
That's why I recommend, if you have access to the raw sequencing data, you'd better try assembly and the binning, the de novo approach, without relying on any reference database. That's what I recommended.
Oh, okay, that makes sense. Yeah. Then the last question we got here today is by Kyle Rourke. Patients taking GLP-1 inhibitors seem like a population that could significantly benefit from precision nutrition. How do you think drugs that regulate metabolism may complicate the prediction methods you mentioned here? Would it be as simple as training these models with new raw data, or would it be much more complicated than that?
Well, that's a great question. I really hope we could have this type of data with, with the drug intake data as part of the training data, so that we can have a more powerful prediction model. So right now, we don't have that kind of data to train the models. But as I mentioned in one of my slides, for example, in the M node architecture is quite general. The input could be the microbial combination, could be the dietary intake. You can also consider the drugs, right, as part of the input. And you can use that to integratively to predict the metabolomic profile. So definitely, the drug information about the drugs would be very, very important. As long as we could have them, definitely, I highly recommend we should incorporate them into the training of the deep learning model.
Mm-hmm. Yeah, I mean, we are very excited about this, and I thought you're clearly leading very many interesting research objectives here, and other people would love to, should know more about it. So I'm very thankful that you came here today. There's one more question. Are there commonalities in the microbiome of population groups that would allow for creating dietary recommendation for specific groups rather than individuals?
Yeah, definitely. Yeah, this is actually a very hot research topic. Because eventually we might just want to stratify the population into several different groups. I think they even come up with a name about this this kind of a stratification or subtyping. So then different subtype or different subgroups, you can come up with a particular dietary recommendation based on the gut microbial combination, for example.
But we are not there yet.
Many people are working on it.
Okay.
If you just Google this type of research, you might find some published papers.
Okay, cool. So we had a good participation today. So we had, at least 80 people showing up. Now we are dropping sharply while we speak. So of course we're 10 minutes past the hour. That's okay. So thank you so much, Professor Liu, for giving this very insightful talk. It's obviously, not everybody is proficient in mathematics or proficient in deep learning methods. So that was good too. And I thought people are at least embracing that more than they used to. And I thought you did a good explanation, to be honest. I thought it's a good explanation you did with with some jokes. I liked it too. We know your son better than we did before. So thank you so much. And we hope to see you in person at some point. And myself, I might also reach out for more personal questions later.
Sure.
Okay.
Thank you.
All right. Thank you so much, Professor Liu.
Thank you. Take care. Bye-bye.
Bye-bye.